My name is Sadie. I am a third year at the Bren School, and today I'm going to be showing you some preliminary results from part of my research on looking at the climate change impacts on ecological physical dynamics in the area from Polynesia. It's a really beautiful island in South Pacific. Specifically, I'll be talking about the influence of hybrid variability on coral reef dynamics in the reef. So, as I'm sure everyone in this room is very aware, coral reefs are incredibly vulnerable to climate change impacts, primarily because corals' response to heat stress is to expel the symbiotic algae from their tissue, which essentially causes them to starve without the energy from photosynthesis from that. This is what we call bleaching, and is a huge, obviously, issue as climate change progresses. However, the impacts of bleaching and mortality on corals is not homogeneous. There's not one single threshold of temperature above which all coral can die. These two photos here are from two different reefs really close to each other in the lagoon of Marina. They're in the same habitat, same depth, very similar situations overall. But as you can see, the coral reef on the left is looking absolutely fabulous. There's tons of different kinds of corals, huge corals in the back. That's a really healthy, thriving reef. But the reef on the right here has undergone a phase shift to an algae dominated state. You can see the structure that these corals have built, but they have since bleached and died and are now covered in that algae. And so there's a lot of different factors that go into determining the corals' thermal stress patterns and the threshold above which they bleach. And the one that I'm focusing on in my research is exposure to high frequency temperature variability. So exposure to high frequency temperature variability can increase the coral's thermal stress tolerance through both genetic adaptation and through short-term climatization at the deep and zero level as quickly as 2 years. And so that's what I'm focusing on here, is this high frequency temperature variability, specifically the daily temperature range of a coral reef. And so this data here is from a global study that took into account coral bleaching data from all across the world, as well as environmental variables such as temperature variability and other thermal stress factors here. And they found that daily temperature range was by far the most influential covariate in bleaching. Just 1 degree Celsius increase in daily temperature range above the average daily temperature range decreased the prevalence of severe bleaching by a factor of 33. So this is a really, really important factor that goes into how overall response to heat stress. And that's why I'm focusing on here. Although the way that this shapes the coral community overall is a little bit harder to determine, and that's where my research comes in. My main research question for this specific project is looking into how varied thermal regimes across areas and lagoon influence coral community composition. And I'm hypothesizing that reefs that experience high daily temperature ranges will show less evidence of past bleaching and mortality, indicated by higher coral diversity, higher percent coral cover, and larger corals overall. And then the objective of this research ultimately is to inform conservation and restoration efforts by identifying populations of corals or locations that might lead to higher thermal tolerance. So those regions could be, or those specific reefs could be useful as donor colonies or nurseries for thermally resilient corals. Oh, there we go. Okay, so the field site that I'm going to and using data from to try to answer this question is the Brea Coral Reef Long-Term Ecological Research Site that a lot of researchers from UCSB and around the world have been collecting data in for 40 years now. And the data that I'm specifically working with is a pretty incredible high spatiotemporal resolution thermistor array, where we have over 40 thermistors in the background for areas of the reef. So you can see there it's really shallow coral reef area inside the lagoon. And there over 40 of these thermistors all at about 1.5 meters to 2 meters depth in consistent habitat surrounding the whole island. So in addition to, of course, spending a lot of time on my computer analyzing data, learning how to code, I also have collected data in the field. And so I've been collecting download data and all of the as many of these thermistor sites as I could get to this past summer, which consisted of boating to the sites as close as I could. Super excited to find thermistors in the water. Some of these sites were insanely hard to get to. and then conducting benthic surveys at each of these sites. And the way that we did these surveys was through transects, or we laid down a transect tape, 4 of them on either side of the banister. And then I took thousands and thousands of photos of all of the coral colonies that fell within half a meter of that transect tape. And we are measuring those coral now in its shape. And then I also took a photo of this photograph frame at every single meter along those transects. And we are categorizing the benthic cover as well as the coral in number of diversity using a machine learning program called CoralNet. And then also in the field, we wrote down the broad benthic categories under each of these cornets. So whether it was coral or alligators. So let's jump into some results. Before talking about the ecological impacts of these temperature patterns, of course, the first step was categorizing these patterns in great detail. So we started here by just comparing the temperature regimes across all of these thermistor sites. So each site here is one of the thermistor sites in the lagoon. But on the left, you see the average daily temperature. There's not even, right? There's only a couple of tenths of a degree Celsius difference between the lowest or the coldest and the lowest site. But we do see a pretty decent difference in average temperature range. So the lowest sites over here on each side of the island are down of about half a degree Celsius on average across this whole data set. Temperature range each day. And then on the northwest side of the island, that's really high daily range sites, and that's up to almost 1.5 degrees Celsius. And so there's, and from that first study that I talked about, we do know that a degree Celsius variation in the temperature range is really significant. That could vary dramatically increase the further results of the corals in those higher daily range. So let's focus in on the highest daily range site and the lowest daily range site. So look at what these temperature patterns are really looking at in more detail. So the first step to look at these temperature regimes is this wavelet transform method. And so I'll break this down a little bit for you here. There's a lot going on, but wavelets are a way to decompose a time series into a sort of time frequency space to figure out the dominant time frames where variability is highest. So we're going to see the strongest signals of temperature variability. And then how do those signals vary over time? So each of these plots is a time series of the 3 years of data available for these sites. The top panel is just a time series. We can already see that the high daily range site has a lot more variability going on. That's really going a lot more at the low daily range site. And then this middle panel here is the normalized range of power state. And so that's telling us the distribution of the signal power. So how variable is that temperature across different scales and frequencies. So our y-axis is hours, and we get into our time period over here. And so the yellow in that middle panel is showing the stronger signal. So you see that kind of yellow dome, that's going to be our annual signal. This is a big temperature change annually. And then this 24-hour and this 12-hour band of yellow, that's what I'm more interested in. That's that high frequency temperature variability on the daily time scales. And you see that those signals are much higher in the high daily range, which makes a lot of sense. That's our high daily range, so that's good to see. And then this bottom panel here is telling us the average power of that signal in the daily time range is what I've selected there, the 16 to 32 hours across this series. So this is important for identifying interesting time periods within this data set where there's higher amounts of variability. So those are going to be some real interesting time periods here to investigate in the high daily range site. Not so much in the low daily range site, but will be interesting to try to figure out why high daily range site is so much variable. Which are the next steps of this part of the project is trying to take those interesting time frames there and fit them into the more regional climate oceanography picture to try to explain that local level variability and those things with regional scale patterns such as eddies or the prevailing currents or other interesting events. But let's jump back into the ecological impacts here. So these are those photos from the beginning, one healthy reef, one not so healthy reef. And these are actually photos from one of the high daily range sites and one of the low daily range sites. So just collecting this data, it really does seem like one of the hypotheses is correct, that there's more diversity, more coral, more live coral at this high daily range sites. And the initial analysis of my coral of the corners data set does support that as well, although it's not the best R-squared value ever. But we do see percent coral cover of hard substrate, but it does seem to be increasing and would increase in temperature. So this is heartening for me to see now. Of course, it'll be awesome to see what this changes as I add in all of the data and all of the photos that we still have to analyze. But it does seem that this is a positive relationship with this covered with increasing in range. And then thinking about how the size structures of these corals are different between the different ranges. It does, so here we come with SIN sites from the wavelets, the highest and the lowest range sites. And the lowest range site does have a lot more coral colonies overall, but they're all a lot smaller. So you see that average size of the specifically else on the high range site. And there's fewer corals overall at the VW site, but they're all a lot bigger. So we have some over here over 2 meters in their longest dimension. That is absolutely massive. And so we have really different distributions of these sizes at each sites. And these are just two of the sites. It'll be very interesting to see what this looks like once we add in all the rest of the sites that we're still mentioned in the show. But in conclusion, these initial results do suggest that a high vivid range does lead to higher percent coral cover in larger corals. But this could be due to a lot of factors. So I have a lot of work ahead of me to try to make sure that I'm isolating the temperature range as a variable, rather than all of the other things that are different around this island and everything else that goes into stress. With that, I will thank everyone who has helped me through this project. A huge shout out to everyone who helped push the field, and also funding to get to the field. So hopefully it's efficient.
Thank you, Sadie. We have time for one question. One question. Anybody really jazzed about corals and heat stress? Yes. Kinsey.
Hi, Sadie. Great talk. I'm curious about the symbionts themselves and whether, like, if they're the same between the different sites, if maybe the high site that shows more resilience also has like greater diversity of symbionts. Maybe that's, yeah, just curious about that component.
Yeah, that's a great question. The genetics of both the coral themselves and the genetics of their symbiont are obviously really huge components of what determines their resiliency and stress. There's tons of work being done on that. I personally am not trained in the genetics part, but there are people at the LCR who are. So that is definitely part of the plan for future work, is to try to integrate some of that research and data that's hopefully, being worked on and published soon, to think about how not only the genetics of the symbiont, but also the genetics of the coral are in these different sites. So that's a great question. We'll see. There's so many factors to take into account, but luckily there are people working on it in various states.
Thanks.